Hi everyone, welcome to our module on clinical trials. A clinical trial is an experiment done with human subjects, and the goal of clinical trials is to determine the benefit of therapy. So one of the ways we know, for example, that certain drugs help patients with myocardial infarction or that certain surgeries work for cancer is because we've performed clinical trials to determine their efficacy. So to understand the rationale behind clinical trials, let's suppose, for example, that we want to know if drug X saves lives. Well, an obvious test would be to give drug X to some patients and then see how long they live or see how many die. And this is actually the way people used to determine whether or not therapies worked. And the problem is that it often suggests that something works when in fact it doesn't. So there are some problems with just giving the drug to people and seeing how they do. The first is that maybe survival or death is the same with no drug X, and we don't know that. We gave drug X to the people, it looked like they did well, but what would have happened if they hadn't received the drug at all? Another problem is the group that's getting drug X knows they're getting a drug. And sometimes people behave differently when they know they're on a drug. Maybe they live healthier lives or avoid bad habits, and this makes them live longer, but it's not due to the drug. Another problem is the investigators know which patients are getting the drug, so they're more likely to take a look at their patients and think they look healthier because they know they're receiving a drug. And the bottom line of both of these second 2 points is that behavior can change when you know you're getting a drug, and this can make something look more efficacious than it really is. So the way we avoid all those problems in clinical trials is by using three techniques. The first is called control, the second is randomization, and the third is blinding. So control means that one group receives therapy and another group gets no therapy, and that group is called the control group. And this ensures that changes in the therapy group are not due to chance. So for example, if we were testing a cancer drug, we might give the drug to one group of patients, but then monitor a second group of similar patients and see how long they live and how well they do. And then we could determine whether the group that got the cancer drug did better, worse, or the same as the control group. The second technique to improve the findings of clinical trials is called randomization. And this is where subjects are randomly assigned to either the treatment arm or the control arm of a clinical trial. And let's think about what that means. If you allowed subjects to pick their arm, you might get bias in the study. Bias is systematic error. Maybe the sicker people would pick to be in the drug group and the less sick people would choose to be in the control arm. By randomly assigning them, you assure that subjects are evenly distributed among the two groups. Randomization means that all variables other than treatment should be equal. So the people in the treatment arm should be getting the drug, the people in the control arm should be getting no drug, but everything else, age, gender, all the other features should be equal between the two groups. And it's very high yield to know for your boards that randomization eliminates a couple of particular kinds of bias. First of all, it should eliminate confounding. Confounders are things other than the drug or treatment that influence outcomes. So they are things like age, weight, or blood levels of certain chemicals. And if they're unevenly distributed, they could influence the outcome. But those things should all be equal in both arms if you perform randomization. And in addition, you should limit selection bias. And as I said before, patients can't choose to be in one arm or the other based on how healthy or sick they are. And in every clinical trial you read, you will see something called Table 1. And Table 1 always shows the demographics of the two groups, the treatment group and the control arm, and it demonstrates that randomization was done effectively. And this is a hypothetical example of what a Table 1 might look like in a clinical trial. So there would be an intervention group right here and a control group right here. And various demographics are shown here to let you appreciate that the groups were appropriately randomized to both arms. So the number of men should be equal in both groups. And here you can see there's 49% in the intervention group and 51% in the control group. There's often a p-value reported to show that there is a non-significant difference between the two groups because they're usually not exactly matched, they're slightly different, but the p-value should indicate that those differences are not statistically significant. Same thing for mean age here is very close. There's no significant difference. Percent that are African American, systolic blood pressure, and you'll see many, many things listed in a table 1. And like I said, if randomization was done effectively, there should be no significant differences between any of the groups. The third element of a clinical trial that improves the outcomes is called blinding. And this is when intervention subjects are given the therapy or drug, but control subjects are also given something called a placebo. A placebo is something that looks like the therapy or drug, but doesn't have the medication or doesn't have any benefit. So in medication trials, usually the placebo is a pill that looks just like the drug, but it doesn't have the medication inside. When you do the trial this way, it means the subjects don't know whether they're getting treatment or not, and therefore they can't alter their behavior based on whether or not they think they're getting the drug. Trials can be single blind when the subjects in the trial aren't aware. Sometimes they're double blind. This means the subjects and the providers are unaware. So if the providers know who's getting the drug or not, this may affect the way they record findings like physical exam findings or symptoms. But by blinding the providers, then you ensure that they can't develop bias when they're recording how patients are doing. And then triple blind means that subjects, providers, and data analysts are unaware. So in some studies, there's concern that the data analysts could skew the data one way or another if they wanted to come out a certain direction. But if it's triple blind, then even the people analyzing data don't know who got the drugs and who didn't. They just analyze the data as group one and group two. And only after they report their findings do you reveal which group got the drug and which group got the placebo. You should know that the best evidence for the efficacy of drugs or radiation therapy or surgery comes from trials that are randomized, controlled, and blinded. So you might ask yourself, why not do these for everything? You're going to encounter many treatments in medicine that do not have randomized, controlled, blinded studies to support them. Why don't we do these for everything? Well, the first answer is because it takes a long time. You have to recruit patients and sometimes follow them for many years to see whether one group does better than another. All this costs a lot of money. So unless there's a pharmaceutical company behind it or a big grant from the government, it's hard to have enough funding to do a clinical trial on therapies. Another problem is the studies take so long to do that sometimes by the end of the study, new treatments already have emerged. So we're hesitant to do these on things when we think the landscape for treating the condition is changing over time. And another reason why we don't have randomized controlled clinical trial data for every therapy in medicine is the parachute example. There are no clinical data showing that parachutes are effective compared to placebo. And this is obviously an example that's cited as a joke. But the point is that some therapies are so obviously beneficial that it wouldn't be ethical to conduct a clinical trial and stick people in the placebo arm, just like you would want to be in the placebo arm of a trial of parachutes and get pushed out of a plane without a parachute. And there are some surgeries that are so beneficial for conditions that are very deadly that we'll never have clinical trials testing them against no surgery. They're just adopted as the standard of care, and we always do them even in absence of clinical trials. It's high yield to know the way data from clinical trials are reported, and to understand this, let's go through a hypothetical example. Suppose drug X were given to some patients with disease compared to placebo, and suppose the arm of the trial that received drug X had a 30% mortality over 3 years. Suppose the placebo arm had a 50% mortality over 3 years. So you can tell from these data that drug X is very effective. It lowered the mortality rate by 20%. There are several ways to report this. The first way is the most obvious way. That's called the absolute risk reduction. It's simply the difference between 50 and 30% or 20%. The second way is called the relative risk reduction, and this is where we take the difference between the two arms, 50 minus 30, and then we divide by the placebo group mortality rate of 50%. So the relative risk reduction for this trial would be 40%. And you might wonder why we would report a relative risk reduction. Why not always report the absolute risk reduction? And the reason is because the relative risk reduction is higher. People like to report this in trials because it makes the effect look bigger. Sometimes the absolute risk reduction will be very small, maybe 1% or less, but the relative risk reduction will be much higher, 30 or 40%. So reporting that number looks better. And so people like to report relative risk reductions. But when you're reading clinical trials, you need to be mindful of that and also notice what the absolute risk reduction is. A final number that trials like to report is something called the number needed to treat. And this is the number of patients that would have to receive therapy in order for there to be a 100% chance of saving a life. So recall that drug X lowered the mortality rate by 20%. Well, we want to know how many patients have to get treated for there to be a 100% chance that a life will be saved. Well, that's going to be 5 times 20%. And the way you do this mathematically is you take the number 1 and you divide by the absolute risk reduction. So our absolute risk reduction was 20% or 0.2, and 1 over 0.2 is 5. And that means that we would need to treat 5 patients in order to be sure we'd save one life. The reality is when you treat one patient, there's a 20% chance you save a life. 2 patients, a 40%, 3 a 60, 4 an 80. But if you treat 5 patients, then there's a 100% chance that at least one life will be saved. And that's the number needed to treat. Now let's talk about meta-analyses. So a meta-analysis is a study that pools data from several clinical trials together. This is an attempt to increase the number of subjects and controls in the two arms and increase the power to detect a difference. So imagine you had a number of small studies of drug X, and they showed that drug X was slightly better than placebo, but the results were not statistically significant. Well, you could pool all those studies together. This would increase your statistical power, and then you might be able to detect a statistically significant difference in outcome between drug X and placebo. The problem with meta-analyses is that the pooled studies are often different. Sometimes they use different patient selection criteria, sometimes they administer the drug or therapy differently, and sometimes one or more of the studies has some selection bias. So the meta-analysis is imperfect, but it is a way to try and get more statistical power out of smaller studies. Finally, in this module, let me talk about the way new drugs get approval. So imagine that you have a drug which appears to work in animal studies and you think it may be efficacious for a disease. What you're going to have to do is conduct several phases of clinical trials in order to receive approval from the government to sell the drug to patients. So the phases are numbered, and you start with something called a phase 1 trial. This is a very small clinical trial, usually with healthy volunteers. They get a small amount of the drug, and what you're trying to do in phase 1 is just determine safety, toxicity, and pharmacokinetics. The key thing to remember, especially for board questions, is that phase 1 involves healthy patients. The only exception to this is cancer drugs and some drugs with a lot of toxicity. We don't want to give those to healthy people, so we'll ask for volunteers who have cancer to receive the drug in a phase 1 trial. But other than these exceptions, phase 1 is generally a trial done on healthy patients. Phase 2 comes next after the phase 1 trial is done. If it looks good, you'll test the drug on a small number of sick patients. And this is where you look for things like efficacy and dosing and side effects. These are often placebo-controlled and often blinded studies, but the key thing here is that phase 2 trials are small because you're still not sure at this point whether the drug is safe or even efficacious. Phase 3 are the big clinical trials that are done before a drug hits the market, usually a large number of sick patients, usually lots of patients in many different centers. These are usually randomized, and the key thing you want to know here is how effective the drug is compared to placebo or whatever the standard care is. And if your phase 3 trial goes okay, then you can apply for and potentially receive FDA approval. And you can imagine that all this testing takes a lot of time and costs a lot of money, and this is why it is generally large drug companies that are able to bring these things to market. It's very hard for a small researcher to do this on their own. There is something called a phase 4 study, which is a post-marketing study. This is done after the drug's been brought to market. And this is done to monitor for long-term effects that maybe weren't detected in the phase 1, 2, or 3 trials. So sometimes we notice after a drug's gone on the market that patients are developing a certain side effect. And if that happens, sometimes pharmaceutical companies will be instructed by the FDA to conduct a phase 4 trial to see if there's an increase in adverse events in patients taking the drug. Also, sometimes you find that when a drug comes to market, it's being used in different groups of patients than it was tested on in the first 3 phases of the trial. So you'll sometimes do a phase 4 study to see the benefit in a different group. But the key thing to remember here, especially for board questions, is that a phase 4 clinical trial is done after the drug's been brought to market. And that concludes our module on clinical trials.